Hi guys, welcome to lecture 2. We're going to talk about genetic drift in this section of our evolution notes. So genetic drift is when in evolution it occurs when random changes or chance events drastically change the allele frequency of a small population. So if the population is large, then the random events wouldn't really significantly change that allele frequency. And we are going to look at two kinds of genetic drift. The first is the population bottleneck, and this is when a population size basically becomes very small very quickly. So this is usually due to a catastrophic environmental event. It could be overhunting a species to the point of near extinction, or it could be habitat destruction. So when the size of the population is reduced so quickly, the many alleles are going to be lost, and then the genetic variation of the population is going to decrease. So basically, just a few individuals are going to make it through the bottleneck. So for example, the northern elephant seals, they have a reduced genetic variation due to humans hunting them, down to as few as 20 individuals. Luckily, their population has since come back, and now they're at over 30,000, but they don't have as much genetic variation as what they did have before they were hunted. The second one is the founder effect. And this occurs when a small colonizing group doesn't have the same allele frequency as the original. So if the population remains small and isolated, then the traits are going to occur at a different frequency compared to the original. So for example, there is an Afrikaner population of Dutch settlers in South Africa, and they are descended mainly from just a few colonists. And today, their population has an unusually high frequency of the gene that causes Huntington's disease, because the original colonists happened to carry that gene with an unusually high frequency. So it's important to remember that the allele frequencies of the new colony do not match the allele frequency of the original population because of the founder's effect. So let's talk about a big thing in the AP is called the Hardy-Weinberg equations. So these equations are used basically to determine if a population is evolving or not. And they also help determine the allele frequency of a population at different times. So if the allele frequency, if those frequencies change, then evolution is occurring. If the frequencies do not change, then evolution is not occurring. So here we've got the equations. P plus Q equals 1, and then P squared plus 2PQ plus Q squared equals 1. So let's break those down. So P, this is going to equal the frequency of our dominant allele. So for instance, if the frequency of the B allele is 0.8, it's going to be 80% of the alleles are dominant. The Q is going to be the frequency of the recessive allele. And basically, the frequency of the recessive allele, if we said was 0.2, or we could say it's 20% of the alleles. You will notice that the decimals and the percents are always going to add to 1 or 100%. Let's look at the second equation. So p squared is going to be the frequency of the homozygous dominant individuals. So in this case, we're going to say 30% of the population has 2 big A's, so homozygous dominant. The q squared is going to be the frequency of individuals that are homozygous recessive. And we're going to say 20% of the population has these recessive genes, the genotype of little a. And then we have 2pq, and this is going to be the frequency of the heterozygous individuals. And we would say that the big A, little a make up 50% or 0.5 of the population. And again, remember that decimals should add up to 1, or if given percents, it should equal 100%. I also want to point out to you that these equations, they are on your equation sheets. However, for the AP exam, you have to know what each of these means. So make sure that you are familiar with them on the AP exam as well as our test. I don't know if highlight the whole area, but make sure that you know them. So let's look at some questions, some examples, and how to break it down. So we're going to approach it as what are they giving us and what do they want? So in example 1, a population with 2 alleles, big A and little a, the frequency of the allele is 0.3. What is the percentage of the population that is homozygous for this allele? So they are giving you, in this instance, they are giving you Q. So they gave you Q, and they want you to determine the Q squared, which I'm sorry, it's just going to show up next to that. I can't do exponents without some difficulty. So in other words, they're wanting you to find out little a, little a. So example 2, in a population of certain frogs in which the allele for brown skin is dominant to the allele for green skin, a drought is going to lead to selection against the green skin frogs. When the drought ends, 12% of the remaining frogs exhibit the green skin phenotype. Calculate the frequency of the dominant allele. So in this case, they have given you Q squared, and they want you to find the dominant allele. So they want you to find P. So in example 3, a population that is a Hardy-Weinberg equilibrium, the frequency of the dominant allele that codes for the tongue rolling is 0.4. What is the percentage of the population heterozygous for the allele? So the 0.4, they told you that it is dominant, so they just gave you P. They want to know the heterozygous, so in this case, they want you to find 2pq. Example 4, in peas, a gene controls flower colors such that R equals purple and little r equals white. In an isolated pea patch, there are 36 purple flowering plants and 64 white flowering plants. Assuming that Hardy-Weinberg equilibrium, what is the frequency of the purple allele? So in this instance, they gave you 64 and 36. So with that, you can figure out that Q squared equals 0.64 or 64%. They want you to determine what is P. So a tip here is always plug in the homozygous recessive numbers into the equation first. Never start with the numbers related to the dominant phenotype. That's going to help you out, figure it out better. Example 5, you sample a population of butterflies and find that 56% show the dominant phenotype. What should the frequency of the recessive allele be in this population? Okay, so they gave you that Q squared equals 0.44. And how we figured that out is basically it shows us 56% of the dominant, and we know that by subtracting we can figure out that the recessive is the 0.44. What they want you to determine is Q. Okay, so let's talk about solving these. So in example 1, the population with 2 alleles, the big A and the little a, frequency of the allele is 0.3. What is the percentage of the population that is homozygous for this allele? So they gave us Q, which equals 0.3. They want us to find Q squared. So what we're going to do is we're basically going to take that 0.3 and we're going to square it, which would give us 0.9. And we could figure out by multiplying that by 100% that it is 9% is homozygous recessive. In example 2, we can see that the frogs, the drought, 12% exhibit the green skin. Calculate the frequency. So they told us that Q squared equals 0.12. They want us to determine what P is. So we know that P plus Q is going to equal 1. So if we take Q and we do the square root of the 0.12 because it's squared, that equals 0.35. So if we take P plus 0.35 and that equals 1, then we should figure out that P equals 0.65. Okay, example 3. They basically told us that the frequency of the dominant allele that codes for the tongue rolling is 0.4. What's the percentage of the population heterozygous for the allele? And they also told us if the population is made of 800 individuals, how many will be hybrids? So they have given us that P equals 0.4. They want us to find what is 2 PQ. Okay, so the first thing that we have to do is we know that P plus Q equals 1. And we know that P is 0.4 plus the Q equals 1. So we know that Q equals 0.6. Okay, so now we have two parts of the 2PQ. So we can do that. So 2 with the 0.4, we figured out the 0.6. That's going to equal 0.48. And then we would multiply that by the 100%. And that means 48% is heterozygous. So the second part is that they asked you about 800 individuals, how many will be hybrids? So the second part is you're going to take the 800 times that 0.48, and that would give us 384 hybrids. Okay, example 4. So in peas, they told us about flowers, that the purple is dominant. There are 36 purple flowering plants. So they gave us also 64 white plants. So they gave us Q squared. And they want us to determine P. So we know that 0.64, because you would take that 64 divided by 100. So we know that that 0.64 is the little r, so homozygous recessive. So that means that Q is going to be the square root of that 0.64, which equals 0.8. And we know that P plus Q equals 1. So P plus 0.8. So P is going to equal 0.2. And example 5. So we're going to sample this population of butterflies, and we find that 56% show the dominant phenotype. They gave us Q squared, which would equal the 0.44, and they want us to determine what is Q. So remember that we're not going to work the dominant. We want to work the recessive. So yes, they gave us this dominant information, but it just doesn't work whenever you do the calculations. So we are going to take the square root of the 0.44, which is going to equal 0.66. And that is Q. Okay, so another example. The graph shows the change in allele frequencies over time in a population. So the first question is the population evolving? So we should be able to tell by the frequency of the alleles here compared to as we go down that the answer here is going to be yes. What is the recessive allele frequency for generation 2? So generation 2 is right here at 0.2. And we know that P plus Q equals 1, and we just found that the recessive is 0.2, so our answer would be 0.8. C, what percent of the population in generation 2 would show the dominant phenotype? Well, we just found the recessive, so if we do the p squared plus the pq so we did the 0.2 and we square that plus 2 times 0.2 times 0.8, then we are going to end up with 0.04 plus 0.32, which is going to be 0.36, and then we turn that into a percentage, so we're looking at 36%. So D, is the population in Hardy-Weinberg equilibrium at the beginning? No, it is not, because we need to justify, because the allele frequency is changing. And then E, identify conditions that would lead to evolution or the population not evolving. So in order to not evolve, remember there would be no natural selection, no genetic drift, no mutations. That is how a population does not evolve. So let's talk a little bit more about Hardy-Weinberg here. If a problem says that the population is in Hardy-Weinberg equilibrium, it basically means that the population is not evolving from generation to generation. It does not mean that the allele frequencies are going to be equal to one another. So for example, here, in 2005, if we observe and count individuals in a population, and we use the Hardy-Weinberg equation to determine the frequencies, and the big A equals 0.7, and the little a equals 0.3, and then in 2010 we do the same thing, and we are at 0.7 for the little a, and we are at 0.3 for the little a, then we would say that this population is Hardy-Weinberg, is in Hardy-Weinberg equilibrium. So there are certain conditions that have to be met in order for a population to be in Hardy-Weinberg equilibrium. They must all be met. So I don't know if you want to highlight that. I'm going to. They must all be met in order for that population to be considered in the Hardy-Weinberg equilibrium, meaning that evolution is not occurring. So the population is experiencing no mutations, no gene flow, no genetic drift, no natural selection, no sexual selection, no artificial selection. So at genetic equilibrium, or Hardy-Weinberg equilibrium, no evolution is occurring. But the above scenario never happens, which means that, again, all populations are going to be evolving all the time. And that brings us to the end of Lecture 2. Next time we're going to talk about evidence for evolution.